Welcome to this lecture on microbial ecology and environmental biotechnology. And in this lecture, the following concepts will be covered. We will discuss on the principle of good management with respect to utilizing the knowledge and understanding of microbial communities in environmental biotechnology. And with this respect, we would like to emphasize on a couple of points. The first is the aim for big benefit. The second is develop and apply for more powerful tools to understand microbial communities and follow the electrons. Aim for the big benefit. Environmental biotechnology aims for the biggest benefits it can deliver to the society, and the biggest benefits will come when environmental biotechnology is directed towards the most daunting challenges of the society, that is the water and energy sustainability. These are the two areas which are identified as the also the priority areas to be targeted for environmental biotechnology research. Now, with this respect the big benefit is achieved when waste is conceived as a resource. Now, the waste waters, sludge, residues and other waste of today, which are actually being generated in millions of tons across the globe, must be viewed as resources that can yield renewable water, renewable energy and in some cases other materials of industrial value. Now, as we understand that these large amount of waste, which are mostly contaminating our water bodies, our surface habitats, are biodegradable. Most of them are biodegradable. So, with that consideration that most of the waste materials are biodegradable, the developing countries are likely to have a double benefit from the application of environmental biotechnology in addressing the issue of waste management, at the same time recovering the resources from the waste. Now, the uncontrolled release of the organic matter, different type of organic pollutants, constitutes serious threats to water quality and human health, and as I said that is true for the entire globe. And any kind of produce organic matter which is released and is having no control on its fate, mobility within the environment, it can provide important forms of renewable energy if environmental biotechnology reliably transforms them into convenient energy carriers like methane, hydrogen and electricity. For example, there could be some more. So, basically the concept is that the organic matter, organic pollutants which are being produced and dumped and released into the environment can be utilized through environmental biotechnology to reliably transform them into different type of energy carriers, that is energy carriers like methane, hydrogen, etc. Now, according to World Bank report 2020, wastewater is basically not a waste, it is basically to be considered as resource, because wastewater is and should be considered a valuable resource from which energy and nutrients can be extracted as well as additional source of water. So, particularly for the developing countries, it has been suggested that the wastewater can be treated through various specific means, and where environmental biotechnology has very specific and very important contribution to make. The wastewater can be treated to recover the water and recycle it. Wastewater can be treated and during the treatment we get the sludge, and those sludge can be used to generate further energy or may be converted to different biosolids, and those biosolids may be utilized for some other purposes. And also we can recover the important elements like phosphorus, for example, to be utilized as a fertilizer. So, this list is actually huge. So, it is only depending on the technology that we are able to develop and implement to treat the wastewater and convert the waste materials into different type of resources. And while we try to do that, while we try to aim that, there is a very big role or very critical role played by the microorganisms, microbial communities, because it is the community, that is microbial community, that is going to work there. So, the previous point that we were earlier discussing, that work for them so that they work for us. So, we need to understand the microorganisms when they work within a wastewater treatment or in a kind of a nutrient recovery system, so that we can make the best use from them. We can recover the nutrients, we can recover the energy or recover the water by utilizing the suitable microbial community function. Now, as it is mentioned that for the developing countries, it is possibly going to offer double benefits, because it will allow us to capture the energy value and also eliminate the pollution hazard. Now, developing countries have the potential to gain a double benefit by investing on environmental biotechnologies that simultaneously capture energy value and eliminate pollution hazard. Now, organic matter that provide important form of renewable energy, if environmental biotechnology can transform them into the different energy carriers. For example, the reduced organic matter, which is basically considered as a substrate for microbial metabolism. So, ideally microorganisms can be used, can be developed, so that the microorganism can utilize this particular material, and here we have the microorganisms, and these microorganisms will metabolize these organic matter, and during this metabolism the electrons will be extracted from them, and as the electrons are extracted, because the organic matter are going to be oxidized. So, they are going to be oxidized during this metabolism. This part of the metabolism, possibly you are aware of that, these are called catabolism. So, during this oxidation process, the reduced organic material are going to be oxidized and the electrons will be released. Now, these electrons will flow and eventually it will allow the organisms to generate energy, energy in terms of ATP, energy in terms of reducing power like NAD pH or NAD HH plus, and essentially it will allow huge growth of the cell. So, that will allow the formation of huge biomass, and the reduced organic matter like the reduced carbon like glucose for example, will be oxidized to carbon dioxide. So, the carbon which is present as reduced carbon in the glucose will be converting to carbon dioxide. Now, if we look at the other part of this particular process, that if we are able to redirect some of this electron flow, that instead of going towards the biomass formation or instead of being oxidized completely in presence of oxygen, if we are able to channelize these electrons to appropriate electron acceptor or to appropriate metabolic processes, we might get energy carrier like methane or hydrogen. Similarly, the other part of this process is that as we are able to develop the microbial system which are capable of oxidizing this organic matter and converting them into methane, hydrogen or other energy carrier, as we are able to utilize more and more organic matter for that process, which otherwise was basically dumped, the reduced organic matter otherwise would have been dumped, would have been released in an uncontrolled manner, and when they were released in a natural environment, whether in a water bodies or in other landfill sites etc., they constitute serious health problems, they constitute serious threats to the animal, plant, human, including human health and well-being. So, this uncontrolled release can be controlled easily as we develop technologies which will be able to convert this organic matter into some electron carriers. There may be some biomass produced, but methane, hydrogen and other electricity kind of things which will allow us to generate huge amount of energy from them. So, it is going to be a double benefit, because in the one way we will gain the energy or energy carriers, on the other way we will reduce the uncontrolled release, and that uncontrolled release otherwise would have be responsible for deterioration of the environment and also impact in human and animal and plant health very severely. So, convenient energy carriers for utilization of organic matter. So, if we see that while utilizing the organic matter through different microbial metabolism, so a number of energy carrier are possible, that basically includes the biomethane to bioethanol and biohydrogen biomethane mix or biohydrogen alone and bioelectricity through microbial fuel cell. So, there are extensive research and there are huge opportunities are being created in this respect, that how the waste material like the organic waste in particular can be treated, can be converted through microbial systems, microbial processes, where microbial communities are actually involved in most of the cases. The second point is the develop and apply more powerful tools to understand the microbial communities. This is very challenging and very important also. Now, when it comes to understanding the microbial communities, we have been discussing about microbial communities and their how we study them to some extent, like we talked about the self assembly etc. But if we look at the different type of methods which are being conventionally used in environmental processes or environmental related processes, it has been based on different isolation of bacteria, which we called cultivable or cultivable approach based processes for a long period of time. So, we generally try to isolate the bacterial culture and try to grow them, and then we try to investigate their properties. So, all in almost all cases what we find that we try to basically grow a couple of bacterial cell from any kind of wastewater. So, suppose we have some waste material, wastewater, if we have some, then from this wastewater we can have a few bacteria, and these bacteria can be tested in terms of their suitability with respect to any environmental processes. Now, that practice has been changed in last few decades, because what we have learned that most of the organisms present in any kind of environment, so including wastewater, are not cultivable. Not cultivable. What does that mean? That means that if we take the water or the waste and try to isolate bacteria from that, out of 100 bacteria present in the waste, if we assume that there are 100 bacterial cells present, only one or even sometimes less than one bacteria will grow on laboratory condition. This we have learned that many organisms are not cultivable. That means 99% organisms are not cultivable. So, 99% nearly it may vary to some extent. So, 99% of the bacterial species or bacterial members present, organisms present in any kind of environmental sample are not cultivable. So, our efforts to understand these and our efforts to address these issues enabled us to develop methods to some extent, and we have developed, the scientists were able to develop what you called cultivation independent molecular method. So, where in cultivation independent molecular method we are not using any kind of laboratory based culturing method. Rather, what we are trying to do, we are trying to use the metagenome, which is basically representing the entire nucleic acid pool, and this metagenome, what we call metagenome, the term the metagenome is coined in order to represent the entirety of the system. So, basically from the water we extract the metagenome. So, that is collection of all genomes All genome means all species and all species genomic DNA, that is together it is called the metagenome. So, in a very simple term. So, collection of all genomes are obtained when we take kind of an approach where the total nucleic acid is extracted from the sample. So, from a wastewater or a system of environmental relevance, we extract the metagenome and then a number of cultivation independent strategies are used. So, we are not going to discuss these cultivation independent molecular strategies over here, but we may talk about them at some other point of time. Now, the another important aspect is that once we started utilizing this cultivation independent method, we have a kind of a what is called as a paradigm shift. Paradigm shift in the in sense that you say that you know that now that only 1% or less than 1% organism was or were actually cultivable. So, all our knowledge based on how these organisms actually function with reference to a particular environmental setup, particular function was based on few cultivable bacteria which we were able to isolate. But as soon as we realize that no, there could be improved methods, and utilizing those improved method, we can actually look into the total microbial community as much as it is possible. So, we made the great advancement with respect to that. So, we will talk about those advancement and how these things are actually done in some other lectures, but what is realized that we need to have more powerful tools, because even after knowing some of the species member or many of the species member which are present in a given environment, a given environmental setup, and with having some idea about how these organisms could be connected to a specific environmental function, it is not yet complete, because we need to win that as we said, it is a win-win technology. Win-win technology in the sense we need to understand completely the microbial process operating in a particular environment, maybe it is wastewater treatment, or maybe it is a removal of a particular pollutant, or generation of energy or nutrient elements from something else. So, we need to have more improved tools and approaches that still those are required to gain a better insights. Now, these are some of the methods that already been developed in last decades or so, following the discovery of ribosomal RNA molecule as a molecular marker for microbial phylogeny and microbial identification, and also having the DNA based methods where, or RNA based or nucleic acid based methods where the cultivation approach can be totally bypassed, and we can straightway hit to the question that what are the organisms present, what are their functions, and how the functions are actually realized, the fundamental questions that we initially asked with respect to microbial community. Now, for example, the hybridization with after the DNA RNA extraction, particularly targeting different ribosomal RNA, taxonomic group specific ribosomal RNA can provide us information on phylogenetic identity of individual strain or coherent groups. Now, for example, if you are interested on phototrophic microorganisms with respect to carbon dioxide fixation process. So, if we know that carbon dioxide fixation is driven by a certain type of organisms. So, if we are able to make the probes small oligonucleotide fluorescently labeled molecules which will be useful in hybridization process, and then following an RNA extraction we can hybridize with those selected probes which are specific to the autotrophic bacteria. For example, we will be able to find out that whether these autotrophic bacteria are present, they are not, and by using different types of probes which are targeted towards different taxonomic or phylogenetic groups, we will be able to actually decipher the different autotrophic population, for example. Similarly, fluorescent in situ hybridization is another technique by which the spatial relationship, because under the microscope we will be able to see that if we have a sludge sample, if we have a biofilm sample, if we have some other kind of environmental sample where we want to see exactly what type of organisms are located where, it is to some extent useful for having this self assembly structure as well, to some extent, not completely, but we will be able to have some idea about who is closer to whom, because if we have a different type of fluorescent probes for different taxonomic group or phylogenetic group, we will be able to find out under the microscope, because here the sample will be treated directly or seen directly under the microscope. Denaturing gradient gel electrophoresis is one of the methods which was very popular with respect to the fingerprinting of the community, and we can use any kind of specific gene, taxonomic marker gene or functional gene, and can assess the nature of the community in terms of a kind of a fingerprinting, or we can also assess the phenotypic potential and the variation within the functional genes. That indirectly will give us some input about the phenotypic potential, because the variation of the fingerprinting or the variation of the DGG result will help us to understand how the phenotypic potential is varying, particularly if we have a treatment, different treatments are given to a community to understand under which treatment condition what type of changes are happening in a particular system. Cloning and sequencing of particular gene, quantitative PCR will provide the quantitation of the community structure, community major member. Reverse transcription PCR will provide us the community function according to the expression of a targeted gene. And finally, the metagenome based tools that which we just briefly mentioned in our earlier slide, that the total nucleic acid based technique, where it is not principally a PCR, may not be a PCR based technique always, and which will provide us the phylogenetic identity of the entire community and will also enable us to get the genomic details. Now, as we continue discussing the particular subtopic on develop and apply more powerful tools to understand microbial communities, we need to understand that in spite of considerable developments in molecular approaches, the existing tools remain inadequate. Still, it is not sufficient. Elaborating the complexity of microbial ecosystems is a very big challenge, because it is not only the fact that we identify how many species are there and what is the relative abundance of how many species, each of the species, but the actual real complexity, the assembly process, the clustering, the interactions, the antagonistic interaction and the synergistic interactions, all need to be resolved, and with respect to the different type of nutrients, the different type of changing environmental conditions, so that we can control them. And also, when you talk about supporting the design and operation of innovative process in environmental biotechnology, so the existing tools are found to be highly inefficient. Now, new molecular tools for microbial community analysis must have higher throughput, so that it is going to analyze a large number of sample at the same time, and also the depth of the information, because many species will be there and some of the species will be relatively less abundant than the other, and it must be quantitative, and it should reveal the structure and function parallel, because we need to know who is there, and at the same time the quantitative evaluation about that, and also their function, that what type of functions are being carried out by them. So, these are again connected to the 4 basic questions that we originally asked, but it has to be more quantitative with more throughput and with depth of information. Now, to do this, new tools are being developed which expands our understanding beyond genomics. So, during our journey from typical laboratory isolation based pure culture based methods to cultivation independent method, we had the invention of the whole genome sequencing technique for microbes, and a number of bacterial and archaeal and fungal strains are being sequenced, and we came to know a lot from those genome sequences, and this is a part of a genomics. But genomics is done only based on the organisms which are mostly isolated as pure culture, although there are techniques where organisms can be sequenced in terms of their whole genome without culturing them always, but those are not regularly available or regularly used in environmental biotechnology setup because of the lack of the knowledge or maybe access to the technology. So, the next one was the metagenomics, where entire population, entire community members, all the members of the community were subjected to the analysis, that is called the metagenomics. Using the total DNA, we are able to do this, and we are able to answer the 4 questions which are related to the microbial community function. Now, beyond metagenomics, so we also have the next couple of points which are basically the metatranscriptomic and metaproteomics and essentially the metabolomics. So, with respect to these, so I will write here. So, we have the metagenomics, which will take care about the total community, and will answer the question who are they kind of question in the first question, that what type of organisms are there. But at the same time, transcriptomics will allow us to access the kind of gene expression, what type of genes are being expressed over there. So, we will be able to know what type of genes are being expressed, and that will help us essentially to answer the question that what is the actual function being carried out by the organisms, because from the kind of genes which are being expressed, if we study that and under different conditions, that will be immensely useful. The study of the proteomics, which will allow us to identify different proteins, enzymes, characterize them and quantify them and relate them to the different cellular function. And finally, the metabolomics, where the study of the metabolic intermediates or cellular functions can be investigated. Now, in recent years or recent decade, we will see, I will say that some more advancement, some very specific and interesting improvements or scientific methods have been developed. Two of them are stable isotope probing and nanosims. So, despite progress in applying the molecular tools like metagenome or metatranscriptome to analyze the microbial diversity of intact assemblage, our understanding of how individual microbial taxa affect ecosystem processes like element cycling remain very weak. So, stable isotope probing, which basically allows the incorporation of stable isotopes of a specific substrate which is under investigation, and the community is fed with that substrate, and later as the substrate is utilized by the community members, some community members will be more active towards utilizing that substrate and some may not be. So, some cells will be preferentially loaded with heavier isotope, and when we do a DNA extraction and a cesium chloride density gradient centrifugation, we will be able to separate out the heavier fraction of the DNA, the metagenomic fraction, and when we sequence and analyze that heavier fraction of the metagenome, we would be able to identify what are the organisms, who are the organisms who are mainly responsible for utilizing the particular substrate. The other one is the nanosims, where nanoscale secondary ion mass spectrometry is used, which is a powerful tool for revealing the element distribution in nanometer scale resolution and elucidating the substrate utilization by a particular cell. So, even a particular cell level in only stable isotope probing, we are unable to define whether a particular cell is doing that. It will come as like a total community DNA, which is heavy DNA, but in case of a nanosims, we will be able to attribute that whether a particular single cell is performing in terms of utilizing a particular substrate, which is again of course level with the heavier isotope. And if we couple this with a fluorescent in situ hybridization or some other method, we will be able to answer even more questions with respect to that. Now, the next important point is follow the electrons. Now, the microbially catalyzed reactions that define the process are usually oxidations and reactions, and as I mentioned earlier, when organic compounds are available for microbial metabolism, microorganisms oxidize that substance or substances and the electrons are released. These electrons are transferred to electron acceptors. If oxygen is there under aerobic condition, these electrons will go to the oxygen and it will create the proton motive force, that will electrochemical gradient, and that gradient will allow the cells to generate ATP and also some form of other form of energy during the course of the metabolism, which are the reducing equivalent. Now, these reactions are basically oxidation reaction and reduction reaction. On the one hand, the substrate is oxidized, like the glucose or the organic matter is oxidized, and on the other hand, the electron acceptors are going to be reduced or reduced. Now, often with respect to biodegradation of pollutants or capturing energy, we often found that these oxidation reactions are very popular, are very common. Now, following the electrons as they move through the microbial ecosystem is the surest way to translate knowledge about the structure and function of the community into practice, because it may not be possible or may not be happening that all the community members, all the species involved or present in a particular community are involved in the oxidation process. It might be possible that out of 1000 species members, only a handful like 50 or 100 or 200 species are actually involved in the oxidation process. Rest may be involved in some other processes. So, how the electron is flowing through the community members would be a very important step to translate the knowledge of ecosystem function into a kind of a practice. For example, the complete oxidation of glucose yield 24 electrons equivalents, and as we can see that electrons are produced during this oxidation. Now, the microorganisms who are involved in this oxidation process can send the electrons to one of the several electron acceptors to gain the energy, and it is known that under the aerobic condition oxygen is going to be the preferred electron acceptor. So, the electrons will be sent to the or accepted by the terminal electron acceptor oxygen under anaerobic condition, whereas under anaerobic condition, when we see that anaerobic respiration is happening, alternate electron acceptors might accept the electrons, and it is a very regular, it is very common event. Now, the overall reaction would be the sum of the electron donating and electron accepting half reactions, which will yield around minus 120 kilojoule of energy, which the microorganism can use for synthesis or other energy consuming reactions. So, there will be a huge amount of energy which will be produced that the microorganism will essentially use. Now, if the electrons from the glucose or the reduced carbon compound which is being oxidized are transferred to methane, for example, if oxygen is not there, for example, and carbon dioxide is actually reduced to take the electrons, essentially the methane is going to be produced. And if the electrons are dumped on carbon dioxide to produce the methane and generate energy by different microorganisms, the overall reaction will yield much less energy compared to the previous case where the electrons are being transferred to oxygen, where we see that it is around minus 120 kilojoule per electron equivalent. Here it is only minus 18 kilojoule per electron equivalent, but this will going to produce a very valuable energy carrier, that is biomethane. So, the energy yield might be less for the cells, but for us, for the environmental biotechnologist, we will have the methane which is going to be produced in those cultures, where these may be some different microorganisms surely, because the aerobic microorganisms are not going to do this. This is a different set of microorganisms. So, this is again the allocations of functions. Within a community, you will have some members who are going to oxidize the carbon, and there are certain other organisms who could be utilizing the electrons and producing methane out of it. Now, some of the electrons from the donor, in this case glucose, are invested to synthesize biomass as well. And so, essentially compared to the previous case where oxygen is available, some biomass is going to be produced, but the amount of the biomass produced will be even lesser than the aerobically produced biomass. Now, nevertheless, this biomass which is produced under this anaerobic condition where methane is produced is a storehouse for electrons, because it is a large storehouse for high energy reactions such as the aerobic oxidation of glucose, but it is a small storehouse for the low energy reactions such as methanogenesis, but eventually some biomass are also produced. Now, if we take the example of BOD, which is a very traditional water pollutant, in any wastewater or polluted water bodies we will find organic pollutants are there, and we consider BOD, biological oxygen demand, as simply a misplaced electron donor. Why we say so? Because organic compounds which are basically responsible for this oxygen demand, when it is also referred as the BOD. So, when this organic matter or biologically oxidizable organic matter enters the receiving water bodies, bacteria, there is aerobic heterotrophic bacteria, they oxidize this, and when they oxidize it, they release the electrons, and these electrons are finally dumped on the oxygen, because oxygen is going to be the electron acceptor for them. Now, that consumes the entire set of dissolved oxygen whatever is available in a stoichiometric proportion, more amount of organic matter is available, bacteria act on that, and huge amount more the amount of oxygen will be depleted, and that will lead to basically bacterial growth. So, what will happen? Each electron equivalent removed from BOD and transferred to oxygen consumes 8 gram of oxygen. So, oxygen level depletes very quickly, and this depletion of oxygen level, if it is too much depletion of oxygen level, then it will lead to serious water quality problems, including odor, color, fish mortality and mobilization of metals and nutrients, and it will change the entire microbial or even macroecological landscape of a particular water body, because the dissolved oxygen is very important. Now, oxidized contaminant whatever is present within this ecosystem or this contaminated water take an opposite role to BOD, because they are electron acceptor like nitrate or maybe the chlorate or maybe iron or uranium or chromium, hexavalent chromium. These could actually accept the electrons. So, removing oxidized contaminants requires the transferring electron to them in proportion to the change in oxidation state from the oxidized pollutant to a reduced form, which is harmless. So, basically nitrate, if we can transfer the electron to nitrate instead of oxygen, it will be converting nitrate to nitrogen, dinitrogen, or we can manage actually the problem of with the chlorate pollution also. Now, the key to detoxifying the oxidized contaminant. So, one way we are trying to manage the BOD, and the other way we are also able to achieve the controlling the other pollutants which are basically oxidized pollutant like nitrate for example. So, now the key to detoxifying the oxidized contaminant is supplying a bioavailable electron donor like hydrogen, which is in most of the universal donor for microbial reductions. Now, recovering the energy value of BOD. Now, electrons carry the energy value of organic matter, and transferring them to complex organic matter like to methane is considered as classic means for recovering the energy value, and the combustion of this methane would generate electricity, and that will possibly capture around 33% of the energy content of the original BOD. Now, this can be further elaborated when we route the electrons to not to methane, but to bioethanol or biohydrogen or certain other products, other electron carriers that can be discussed in subsequent classes. So, for this we can follow these two literature, Microbial Ecology to Manage Processes in Environmental Biotechnology by Bruce Rittman, and a Vista of Microbial Ecology and Environmental Biotechnology by Bruce Rittman. And to conclude, the aim for big benefit waste to resources like targeting the major aspects of the recovery from the waste material, sustainable water and energy management is discussed, and it is also highlighted that this could be a double benefit for many countries, including the particularly the developing countries. There is a need to develop and apply more powerful tools to understand microbial communities, more to achieve better understanding, because better we understand the communities, better will be their application. And we also discuss the point follow the electrons, where get the energy and treat the pollutant. So, how this can be translated, it is a big point of interest. With this, thank you.